as well as proteins in general. So what is a protein? Proteins are basically the things that do all the functions in a cell, and they have a lot of forms, and their forms give their functions, so that's how they have a lot of functions. So in the protein structure, they have a lot of different forms a lot of forms of structure that have been categorized. So there's primary structure, which is basically just the actual sequence of amino acids that make up proteins. The secondary is whenever these chains have turned into pleated sheets or helices. The tertiary is whenever it's actually formed into a complete 3D structure. And the fourth is whenever it has multiple 3D structures. Actin is itself generally the most plentiful of intercellular proteins among eukaryotes, and is especially prevalent in muscle cells. It's densest in a region that's called the cortex under the cell membrane, and it's connected to the membrane there by a variety of binder molecules. And the filaments that actin forms can change the shape of a cell by, like adding on to, dismantling, or shifting the parts of the actin cytoskeleton. And mutations in beta and gamma actin genes can cause Barrett's or Winter syndrome, which causes malfunctions in actin. And because of that, it will cause deformities in how the face is structured. So G-actin structure. G-actin is the monomer form of actin, and it has a globular shape, and it occurs when there's no salt and when it's in low concentrations of proteins. So it has a bound nucleotide, usually it's ADP or ATP, and this serves as like a cleft between the two domains, and then within those two domains there's again two subdomains, and then there's minor sites off of those subdomains. So the primary structure of actin contains 374 amino acids, and the tertiary structure contains a multitude of alpha helices, such as here. And there's some pleated sheets, which you can kind of see here a little, with particular note to a 5-chain beta pleated sheet that occurs in both domains. The F-actin structure. So F-actin is the polymer that has an appearance that's asymmetrical and stringy. Here's a reconstruction using, I believe it was crystallography. And so F-actin is the form that's used to compose actin filaments, and they tend to have a helical structure, and is composed of two strands of actin subunits. And it's very dynamic and variable, so it kind of shifts between structural states rather than just being a single structure. And its behavior, so it's got negative ends and positive ends. The negative end is also known as the pointed end, and the positive end is also known as the barbed end. So you can see the negative end, positive end. So the barbed end is composed of the smaller domain of the actin protein, and so that means that more actin proteins can bind onto it. And the negative end is where the binding cleft is uncovered, while the positive side has the nucleotide binding region facing an adjacent subunit. Actin filaments can form into bundles and networks. Bundles are dense and parallel. They're like a group of parallel lines, whereas networks are loose and have filaments overlaid on one another.